Assalamu alaikum and good morning to one and all. Today we'll study about the drug discovery and development process. So let's start with the part 1 of this chapter. So it is very detailed session and I have broken this session into different parts. I am Professor Sheikh Abu Sufyan. Welcome you to my YouTube channel Pharma Learning Forever. At the end of this e-learning session, you are able to distinguish different phases of drug development and discuss drug discovery process in a detail. So let's start with the different phases of the drug development. So the drug discovery and development process, it is broadly divided into two important phases. So first phase, it is a discovery phase where the drug it is just discovered and it is not completely matured to bring that drug into the market. So what is done in case of the discovery phase? So in case of discovery phase, you are identifying or the identification of new chemical entity as a potential therapeutic agent. It is done in case of discovery phase. There are n number of drugs are present. Out of these n number of the drug, which drug it may produce particular pharmacological action or it having potential to produce good therapeutic effect and it works as a therapeutic agent. So all these new chemical entities are identified which having the potential to produce therapeutic effect and to act as a therapeutic agent. Like in case of the COVID-19, now you can see the many scientists, they are working on the existing drug. Like some of them, they are targeting on particular medicine to bring about some existing antiviral drug to produce pharmacological action or against coronavirus. Likewise, some of the scientists, they did work on the steroid and they have brought the use of existing steroid for the treatment of the COVID-19. Likewise, there are n number of existing drugs are available. You can either work on those existing drugs and establish those drugs as potential therapeutic agent, or you can completely work on new chemical entity. You can synthesize new molecule based on the learned mechanism of action. Got it? So this is how we work in case of discovery phase. So although I have written this in just one statement, but the drug discovery phase, it is very detailed phase. It doesn't happen very quickly. It takes time. Okay. So the first phase, it is the discovery phase of the drug. Second phase, it is the development phase. So in development phase, what you are doing, the compound that you have identified in discovery phase as a potential therapeutic agent, that compound it is further developed with the help of different testing in the human models as well as in case of the animal testing. So in those testing, you see that the drug it is safe and drug it is efficacious in nature. In addition to this, you also look into different aspect of the drug development, like you study their pharmacokinetic parameters, ADME profiles, their T-half and many aspects, so that the drug it is more safe and it produces good efficacy in that particular patient. Okay, so the drug discovery process, it is broadly divided into two phases: discovery phase and second one it is development phase. So what I will be doing in today's lecture, so before going ahead with each and every step of the drug discovery and development, first let's see the overview of the drug discovery and development process. So the first stage that as we have seen, it is the drug discovery phase. So the drug discovery phase, it is the step-by-step process. It is not like every time drug it is discovered by the process of serendipity. That is like accidental drug discovery process. You were working on something else. You have got some observation and based on that observation, the drug it is accidentally discovered. So that is called as the serendipity, isn't it? So always there is no possibility of serendipity, but nowadays the drug discovery process, it is very systemic process and you follow the systemic approach in case of the drug discovery. So there are different stages are involved in case of first phase, that is the drug discovery phase. like target identification, where you are identifying the target, hit and lead identification, lead optimization, and pharmacological profiling. So in today's lecture, we'll see each and every stage or each and every step of drug discovery in a detail. So this is the first phase of the drug discovery. Second thing, it is the preclinical development. So first, when you identify any therapeutic target or any therapeutic agent which may having potential to come as a drug or to work as a therapeutic agent, if that is the case, then that drug, before testing into the human being, you conduct study on the animal. So those animal study, it is called as preclinical development. So in animal study, you study different parameter of the drug, like their toxicology. So where you study at what dose it produces the sign and symptom of toxicology, and up to what dose drug it is safe. So that you study in case of toxicological study. You also study their pharmacokinetic parameter, synthesis scale up. Is it feasible to do the scalability of that drug for to suffice the need of large number of population? Is it feasible to synthesize that drug at affordable rate? So that is also study at this point, and at the end you also study the formulation of the drug. What is that formulation which is better for human use? Either that drug can be given in the form of the tablet, capsule, syrup, or the injection. That is studied in the preclinical development of the drug. So this is the second stage, that is the preclinical development. Third stage, it is a clinical development. Now once drug it is identified which having the good therapeutic effect, good pharmacokinetic parameters, less toxic effect, so drug it is good and safe in nature, and you have also identified the formulation that can be given for the human use. And then once you're done with this, then you can proceed with next stage, that is a clinical development of the drug. So in clinical development of the drug, there are different phases. So where you do or you are doing the study for the safety and efficacy of the drug in the human volunteers. So there are three stages are there, like phase 1 stage, phase 2 stage of clinical development, and phase 3 stage of clinical development. So we'll be seeing this in a detail in subsequent session of this chapter. Okay, so third stage it is the clinical development. Fourth stage it is regulatory approval. Now once you have done with this study, you will get certain set of the data. So you will compile that particular data, and based on that data, you're trying to justify, you're trying to convince the regulatory authority that your drug it is safe, it having good efficacy against particular disease. If that is the case, then that data it should be sent to the regulatory approval. So in case of the India, like you can send it to the Food and Drug Administration and other regulatory authorities. So what you are doing at this stage, simply you are submitting the data that you have received by doing the study at the earlier phase, that is the phase 1, phase 2 and phase 3, right? So whatever data you get, so that you are submitting to the regulatory authority. That regulatory authority will review that data, and based on the data that you have submitted, regulatory authority will make the decision. Either the decision could be they may allow you to market the drug, or they may not allow you to market the drug, or they may request you for the additional study on the human volunteers or the additional clinical study, isn't it? So this is how drug it is processed. So this is called as the regulatory approval. So once, now suppose the drug it is approved by the regulatory authority, then such drug are allowed to launch into the market, isn't it? So now if the drug it is launched into the market, it doesn't mean that the process of drug development it is over. Although drug it is launched into the market, but there are some adverse effects, there are some side effects, there are some the new application of the drugs can be understood when the proper surveillance of the drug it is done. So once new drug it's bring into the market, the next stage it is a phase 4 study. So in phase 4 study, what you are doing, you are monitoring that drugs till the point you are satisfied that the drug, it is the safe in nature and it is efficacious in nature. So what you are doing, you are conducting the study that is called as post marketing surveillance. Since this study it is conducted after the marketing of the drug, so this study it is called as post marketing surveillance. So this entire process, on the slide deck you may feel that it is a very simple process, but it is not a simple process. It is a very tedious process. Good amount of investment it is involved. The number of manpower are involved and number of experts are involved in the process of drug discovery and the development. Now if you just look at the time that it consumes in each phase, then you will get idea about how tedious this process is. For example, in case of drug discovery phase, you need at least 2 to 2.5 years, and you may start with 100 molecules in the first phase, that is drug discovery phase. The moment when you move from drug discovery to preclinical development, from those 100 drugs, your 80 drugs are already being rejected. So you have selected only 20 drugs based on the first study, that is the target identification, hit and lead identification, lead optimization and pharmacological profiling, right? Then for 1.5 years you are conducting the study which is related to preclinical development, so animal study. So maybe from out of 100, 20 molecules you are studying for preclinical development, right? So out of these 20, after conducting preclinical study, some of the molecules are rejected based on the data and some of the molecules are selected. So out of 20, either 10 or up to 2 molecules are selected. So this is based on the previous data, and those drugs are selected for conducting the clinical trials on the human volunteers. So this study, it's continue up to 5 to 7 years, and you can see out of 100, the 20 drugs come into the second phase, then 10 to 2 drugs just enter into third phase, that is the clinical developments, right? Once this study is done, again some of the molecules are rejected and only 1 or 2 molecules, their data, it is submitted to the regulatory authority based on the data which is received from the clinical development, isn't it? Now out of these 1 or 2 molecules, the regulatory authority, it may allow both the molecules or it may allow only 1 molecule to launch into the market. Now once drug it is launched into the market, again work it is not completed. The drug, it is under the supervision, so you are conducting the post-marketing surveillance to further ensure its safety and the efficacy, right? So now when drug, if it passes the regulatory authority, so that drug, it is called as IND drug, that is called as investigational new drug, that is the new drug molecule. It is not an established drug, it is a new drug molecules. Since it is the new drug, till the time through the post marketing surveillance you get very good data that indicate the safety of the drug, right? Till that time you are conducting the post marketing surveillance. So once the drug passes post marketing surveillance phase, now that drug, it is called as a drug, otherwise it is a new drug. So new drug, it means it is under the surveillance, and drug, it means it is the established one, it having the good data to support its safety as well as the efficacy. Okay, so this is how the complete process of drug discovery and development is there, and it takes around the 10 to 12 years to take place. Okay, but now due to the artificial intelligence and with the help of many computer aided drug discovery process, nowadays these drug discovery process and the development, it is nowadays not taking these many years. So at earlier phase only you can reject those drugs which are having the less potential to produce pharmacological effect, and this can be shrink to 5 to 6 years. Okay, so this is how you can conduct the drug discovery and development process. So let's move on to the first phase, that is the drug discovery. We'll see each step in a detail. So there are 4 stages, like target identification and validation. Second, it is the hit and lead identification. Third, it is the lead optimization, and fourth phase it is called as pharmacological profiling. So let's see each phase of the drug discovery to IND in a detail. So first phase it is the target identification and validation. So what you are doing here, you are identifying particular target. So disease of interest or the medical need it is identified. So in current situation of COVID-19 pandemic, the most of the pharmaceutical company they are engaged with the research on the COVID-19, isn't it? So the COVID-19 it is a disease of the interest in current situation. Likewise, maybe the disease of interest may change in the future, and there are some disease of the interest. They are always there. So the scientists they are consistently working on those diseases. Like people are consistently working on the HIV. They are consistently working on the new anti-diabetic molecules, anti-cancer drugs. So these are the consistent target where scientists can work. But again, based on the medical need, the disease of interest may change. So first thing that you need to do in case of the drug discovery process, first you need to identify the disease of interest. Then you try to understand the mechanism of that particular disease and its progression. How that disease it progress. What is the mechanism behind their pathophysiology? How it produces the disease, right? So likewise in COVID-19, the people have identified different aspects like angiotensin receptor. Some of them they are working on the corticosteroid. So corticosteroid it is the another side. So this is how you can understand the appropriate target for the drug discovery process based on the mechanism of the disease and its progression. Likewise, you can once you do with that, you can identify viable therapeutic target, which receptor you want to target your medicine, right? So either it is a receptor or it is the enzyme or it is the transport protein. So it is everything it is based on the mechanism of the disease and its progression based on their mechanism, right? So you are identifying basically here disease of the interest, then you are trying to understand its mechanism, and based on the mechanism you identify viable therapeutic target, whether it is receptor, whether it is enzyme or it is transport protein. So once you identify viable therapeutic target, now you can start doing this study on the animal model. So what is that? So that is called as development of a robust biological assay for testing of the compound. So you can identify the particular biological assay for testing this compound. So you can test this on the whole cell, maybe on the whole animal, or you can study this on the transform cell like the cell line, recombinant technology. You can do this study, or you can also do this study on cell-free biochemicals. So you are not using the cell, but you are using a biochemical for testing that particular target, or for testing that particular the target that you have identified. So what is basically you are doing? You are first identifying that target and then you are validating that target by using various robust biological assays for testing the compound, right? So you have identified the target, you have also identified what could be the robust biological assay to validate the target that you have identified. So this is the first stage, that is the target identification and validation. So although it's look like it's a very simple process, but it is not that simple. It is very challenging in nature. So there are various limiting factors are there which limit this process or which reduces the speed of identification of appropriate target. Now what are those limiting factors? So these limiting factors it include emergence of unexpected adverse effect. It may happen that you have identified a particular target and when you test that on the animal model, it is observed that that drug having very severe adverse effect. So the moment you identify the adverse effect to that drug, you need to stop study at that point only. So that is one of the limiting factor in case of the target identification and drug discovery process. Second limiting factor could be the cost. So to perform the discovery process or to do this all testing, it must be economically feasible. So if you are pharmaceutical company or if you own pharmaceutical company, so you also look into your budget. It must be the economical and it fall into your budget. So most of the time, although there are many therapeutic targets are available, but the cost and complexity of the drug discovery process, it is the limiting that particular research. Likewise, the third could be increasing regulatory hurdle. Bringing any drug into the market, it is not a simple task. There are many regulatory hurdles that you need to pass on till the time drug it is launched into the market. Okay, so these are three limiting processes are there, which reduces the speed of the drug discovery process as well as the first stage, that is a target identification. Okay, so I have done with my first part of the session. So before going ahead with the second part, let's have the quick question and answers. So my first question is, what is difference between the drug discovery and process? Yes. So let me correct this question. So I would like to know what is the difference between the drug discovery and development. Yes, Aksa has given the first answer. The drug discovery is the identification of new chemical entity as a potential therapeutic target. Okay, and development phase where you are studying their safety and efficacy. Yes, you are right, Aksa. So new chemical entity it is which having the potential therapeutic target, that is identified in case of drug discovery phase, and drug development phase we study their safety, efficacy and other pharmacological parameter. If that is the case, then that is called as a drug discovery phase. Okay, so we have seen the difference between the drug discovery and drug development, but the answer given by the students are not encouraging. Only 4 to 5 students have given the answer. I am expecting everyone to be very active while giving answer in the chat box for the quiz part. Okay, my second question is, enlist the stages of drug discovery. What are the different stages of the drug discovery? So just enlist those stages of the drug discovery. That is the first phase. Yes, so first phase it is the target identification and validation. So Aksa and Nurun Nisa has mentioned that. The second phase it is the hit and lead identification. Very good. Third phase it is the lead optimization, and fourth phase it is the pharmacological profiling. Okay, so nice to see that you are giving the answer. So we have seen the first phase, that is the target identification. Second phase it is the hit and lead identification. Third phase it is the lead optimization, and fourth phase it is the pharmacological profiling. So out of this, the first three stage will be seeing in detail in today's session. So already we have studied with the target identification, what is done in target identification, so that we have already studied. Okay, so there are total 4 stages are there in case of the drug discovery process. My third question is, what is target identification? Okay, so Nurun Nisa has given the first answer, that is a disease of interest it is identified. Okay, but that is just first stage. Then you identify enzyme or transport protein. Aksa has mentioned that. So identify viable target. Okay. And then once you identify viable target, then what you are doing? As Kavita mentioned that disease of interest, understanding the mechanism and its progression. Yes, that is the intermediate stage of the target identification. Now what you are doing after identification of the target? What is the next stage? Yes, Nida has mentioned identify viable target like receptor, enzyme and transport protein. Okay. So just identification of target is not sufficient. In addition to target identification, what else we are doing? Yes, so Aksa has given the answer, develop and validate robust biological assay. So further validating this target is very important. So to validate this target, what you are doing, you are identifying or you validate a robust biological assay, maybe the whole cell or on the whole animal you can conduct this study, or maybe the cell line or the biochemicals, cell-free biochemicals can also be used to validate your target. Okay, so identification of the disease of the interest, then studying their mechanism and the progress, and based on that you can plan which receptor, enzyme or the protein that you want to target for giving or for studying their pharmacological action. Okay, so that is what we are doing in case of the first stage, that is a target identification. So I have done with my first part. Let's move on to the next part of the session, that is a heat identification. Now what is the meaning of the heat? Let's first try to understand for that, and then we can move on to the next part. Okay, so the heat identification, it is basically process where the compound which assume to response the assay in favorable manner, it is identified. So those compound which assume to response the assay in favorable manner are called as the heat. For example, in case of COVID-19, they have identified the compound which may respond in favorable way. The first the compound which was identified was anti-malarial drug. That was the chloroquine. So they are first identified as that compound. But then it was observed that chloroquine does not having much role to play in case of COVID-19. Then they have identified the some of the therapeutic agent like the steroid, corticosteroid, and it was observed that the steroid, it helps to the certain extent to reduce the transfer of the disease from the moderate to the severe. So it halt a progress of the disease from moderate COVID-19 symptom to the severe COVID-19 symptom, isn't it? So likewise, many drug that you identified, which you assume that it may respond in a favorable manner as a scientist. So all those compounds that you are assuming to respond the assay in a favorable manner are called as the heat compound. So those compounds are called as the heat. Now each set of these heat are tested separately against the target assay to identify validated heat, isn't it? So now once you identify the heat, that is not sufficient, then you are testing against that heat against different target. So the target could be the receptor, the target could be the protein, or target could be the assay, right? The target could be the enzymes. So you are studying there the binding on those receptor, the binding of this target on the enzyme, as well as the different aspects, so that these heat can be further converted into some optimum drug, isn't it? So you are doing proper validation process after identification of the heat. Once you identify heat, this heat can also be converted into the lead. So that is called as the lead finding. So what you are doing in case of the lead finding, suppose you have identified the 100 or maybe the 50 or 60 compound as the heat compound. So those heat compound can be further studied, and then those compound can be converted into the lead. So this process, it is called as the lead finding. What is the lead finding? So lead finding, it is the next stage where you are rejecting few drugs from which are identified as the heat, and you are selecting few drug for further process. So the drug those you have selected after validating the heat, it is called as a lead compound. Okay, so again it is not a drug, or it is not a it is not a new drug, it is called as the lead drug, right? And the process that involved, it is called as the lead finding. So there are two processes are there in case of the lead finding. First process, it is called as the convert heat to lead. So in the short form, we called it as a H2L. So heat to lead. So you have identified 100 heat, and then you are converting those heat into the lead based on the systematic approach that you have adopted. So that could be the one of the way. Second way could be modifying existing compounds. So how you can modify existing compound? For example, there are different series of benzodiazepine drugs. So these benzodiazepine drugs are come into the market after modifying existing basic structure of the benzodiazepine, like diazepam, alprazolam, and many drugs are there in the category of the diazepam. These drugs are launched into the market after just modifying the existing basic structure of the benzodiazepine, isn't it? So this is the second way of the drug discovery, where you are simply modifying existing compound which is available which having good pharmacological action to optimize the properties, either the physicochemical properties or maybe their efficacy as well as the safety. So there are two processes: convert heat to lead through the systematic approach that you have taken, and second one, it is modifying existing compound. So let's see what are these stages, or what exactly doing in all these stages, and how you are further moving ahead with the drug discovery process. So the first process, that is start with the heat and create a lead compound. So it is called as the heat to lead, or it is called as the H2L. So what you are doing in this stage, you have synthesized and test analog of heat to optimize certain properties. What do you mean by analog of the heat? You have identified the drug or chemical molecules which having the same structure. Since it having the same structure, it increases the possibility or probability towards binding towards particular target, because the receptor, it is a protein. Now based on the protein structure, after doing some systematic study, you can predict which drug that may bind to this receptor, and it may activate it or it may produce the antagonistic effect based on your the expectation. Okay, so you have synthesized and tested analog of the heats to optimize certain properties like their biological activity. That maybe you are not satisfied with the existing heat compound that you have identified, maybe it having less biological activity and you want to increase their biological activity. So what you can do, you can try with changing their existing structure, so you may get good biological activity. Or maybe you are also working on identifying the target effect, so you want to improve their target effect, you want to have the more selective effect. We have seen if the drug having the non-selective target, maybe if it's bind to n number of the receptor, then it increases possibility of side effects. And if your drug it's bind to the specific receptor, it reduces those side effects, because now drug is not binding to those particular receptor, right? So you are trying to optimize the property where you are seeing that the drug bind to the specific identified target and it having the less affinity towards other receptor. So this could be the second the objective that you want to achieve when you are converting the heat into the lead. Third could be the selectivity to avoid side effect or the toxicity. So this you are doing specifically to convert heat into the lead. Now heat that you have identified, it may not having the good biological activity, it may not having good identifiable target effect, or it may not having good selectivity towards the receptor. Now when you do the change in their structure, or when you change the structure of that particular molecules, and when you get all these desired property, you get the good biological activity as per your expectation, the drug is binding to the identified target, so that is called as the identified target effect. And drug, it is also selective, it is not having the effect on the other receptor. When you get all this property as expected, now that heat, it is converted into the lead. Now those compound that you have selected are called as the lead compound, and those compound which are rejected, now those are can be further studied, you can further change their property, and again you can try to convert those heat compound into the lead. So this is how research it is going on, and this is the continuous and tedious process. So this, we have seen the first stage, that is the heat to lead identification. I hope you understood this process of the heat to lead identification. So during this process, nowadays you are not using the manual technique, but you are using the systematic approach of drug design, and there are many tools are available. Those tools you can use to speed up your process of conversion of lead to heat. Now what are those tools? So first we'll see what is the objective of using these tools. So you are using this tool to create a novel compound and to create the optimal compound which having the activity as per the expectation of the researcher. So to create novel compound and optimal compound, you are using drug design tools. So we can use the computer aided drug design system, it is called as the CADD. So it is very emerging, and nowadays most of the scientists when they identify the hit, they convert it into the lead after doing computer aided drug design study. So based on only this study, once you do this study, you will get some data that can be predicted by the computer, and based on those prediction you can take certain level of decision. So this is one of the way you are using the tools. In addition to that, you can also go ahead with conversion of hit to lead based on medicinal chemistry experience. So to have this particular experience, you have to be very senior in the field of the drug discovery. So if you are a very senior person, you are working from last 20-30 years in the particular field, then based on that particular experience of your medicinal chemistry, you can modify particular structure. Maybe you can just change one group of the heat, so that that heat can be converted into the lead. Okay, and you get the favorable expected pharmacological action and expected selectivity towards the receptor. So this is one of the way. Second way, it is converting existing compound, modifying the existing active compound. So those are not a heat. So in first stage, what we did, we have converted the heat into the lead. But in this stage or this process, what you are doing, you are modifying existing compound. Now, for example, in case of 5-hydroxytryptamine, you know that 5-hydroxytryptamine, that is also called as a serotonin. So serotonin, its work on the brain, it modulate the mood, right? It having very good, it also having the role in case of the migraine. So the new drug, that was the sumatriptan. So inadvertently I have written it as a serotonin. So I'm doing that change. So the sumatriptan, it was identified by just modifying the structure of the 5-HT, that is 5-hydroxytryptamine. And sumatriptan, it is very good anti-migraine drug. Now, if you look at their structure, only modification it is from the 5-hydroxytryptamine is this OH group, it is replaced by this group. Okay, so just replacing one group, it has converted existing molecule, that is 5-hydroxytryptamine. It is not a lead, it is a neurotransmitter, it is existing molecule. Now that existing molecule, it is converted into the lead by just changing the structure, and this is how the sumatriptan, it's come into the market by changing the structure of the existing molecule. Okay, so this is also another way. So by changing the structure of the existing molecule, what you are doing, you design structural changes and create compound to improve their biological activity and also the selectivity for the target. You are trying to eliminate